Welcome back, everyone. Let's just dive right into it. Today, we're tackling a high-yield explainer focused entirely on microbiology fundamentals. We're going to break down the body's 3 lines of defense to help you absolutely crush your surgical technology certification exam. Before we get totally lost in the microscopic details, let's remember why we're actually studying this stuff. This quote really hits the nail on the head when it comes to the real-world stakes. Decontamination and sterile technique might not seem super flashy when you're just hitting the books, right? But failures in these exact processes lead directly to deadly surgical site infections, or HAIs like MRSA and C. diff. As a surgical tech, understanding this microscopic battlefield is literally your ultimate weapon for keeping patients safe.
So, here's our roadmap for this explainer. We'll cover the first line's structural barriers, the second line's chemical responders, and then test you with a clinical case on inflammation. After that, we'll dive into third-line adaptive immunity and wrap the whole thing up with a case on patient immunity.
All right, Section 1, first line, structural barriers.
We're kicking off the pathogen's journey at the body's outermost fortress, our innate nonspecific protections. And honestly, it's pretty fascinating just how multi-layered these physical and chemical barriers actually are. Take your intact skin, for example. It's not just a physical wall, it's actively secreting lactic and fatty acids, creating this harsh, low pH environment that basically stops bacterial growth right in its tracks. Inside the body, mucous membranes and cilia physically trap inhaled invaders. Meanwhile, you've got lysozymes floating around in your tears and saliva actively killing off gram-positive bacteria. Oh, and don't forget the digestive system. It swings from a highly acidic stomach all the way to alkaline intestines. So the absolute crucial point to remember for your exam and obviously for the operating room is this. The second the surgeon's scalpel makes that initial incision, this critical first line of defense is totally breached.
Moving right along to Section 2, second line, chemical responders.
So what happens once that physical barrier is crossed? Well, pathogens are instantly met by a rapid internal response team. Enter the phagocytes. Seriously, highlight this term in your notes right now. Phagocytes are a specific type of leukocyte, and you can think of them as the garbage collectors of the human body. They actively seek out, surround, and just completely engulf bacteria and cellular debris. For your boards, lock away the fact that neutrophils are the most abundant and efficient phagocytes deployed in this initial innate response.
Okay, you absolutely have to know the sequential mechanical steps of this cellular digestion, which is known as phagocytosis. Step 1 is chemotaxis. Think of this as a chemical alarm bell drawing the phagocyte directly to the bacterium. Step 2, the cell membrane basically folds inward to form a vacuole, called a phagosome, right around that engulfed bacterium. And finally, in Step 3, a lysosome packed with powerful digestive enzymes attaches to that vacuole. This forms a phagolysosome, which totally destroys the invader. It's like a microscopic demolition crew.
Looking at the diagram here, you can literally see how the bacterium binds to the cell receptor first. Then, during that absorption phase, the leukocyte basically swallows the pathogen whole, leading to the final formation of the internal phagosome. And if you look closely, see how the antibody, the IgG labeled right there, is basically flagging the pathogen to kick off this exact destructive process. It's pretty amazing.
Let's shift gears to another vital piece of this second line of defense, the complement cascade. These are a group of totally inactive blood plasma proteins that trigger directly when they bump into substances on a pathogen. What's absolutely critical to remember here is that they're part of the innate response. They do not need antibodies to get started. They just coat the pathogen to make phagocytosis way easier. And the cascade sequence ultimately results in cell lysis. Simply put, it literally makes the bacterial cell membrane burst wide open. Boom, done.
Section 3, clinical case, inflammatory response.
Okay, let's hit pause on the theory for a second and actually test your practical application. Here's your clinical scenario. A patient presents with edema, redness, heat, pain, and purulent exudate, which, is pus, at a healing surgical incision. Think carefully about how your exam will phrase this. What specific response is occurring and exactly what cellular action causes that edema? I'll give you a second to lock in your answer.
Well, if you guessed the inflammatory response, you are right on the money. But let's unpack the why behind all that swelling. The root cause of the edema is that injured mast cells and basophils release histamine and kinins. This release directly results in increased capillary permeability and vasodilation. Basically, the blood vessels widen and get super leaky, and that leakiness allows inflammatory exudate and infection-fighting leukocytes to just flood into the area. That local pooling of fluid is exactly what causes the visible swelling and redness your patient is dealing with.
All right, Section 4, third line, adaptive immunity.
Now we're entering the highly specific, memory-based protections of the immune system. I can practically guarantee that distinguishing between humoral and cell-mediated immunity will be on your exam, so let's break it down sharply. Humoral immunity is driven by B cells. Its entire job is producing specific, circulating antibodies in the lymph fluid and plasma that basically stick to foreign antigens, marking them for destruction. Now, in sharp contrast, cell-mediated immunity, or CMI, is driven by T cells. And the massive differentiator here, CMI does not produce antibodies. Instead, it relies directly on macrophages and cytotoxic T cells to go out, seek, and destroy infected cells.
See this table of the 5 classes of immunoglobulins? I'd highly recommend taking a screenshot right now. Seriously, it's a perfect study guide. I want you to pay special attention to IgE. That is the specific antibody responsible for allergies, anaphylaxis, and immediate hypersensitivity. And definitely don't sleep on IgG. It's the most abundant, it directly attaches to phagocytes to aid digestion, and crucially, it is the only one that crosses the placental barrier to protect a developing fetus.
So another massive part of adaptive immunity is artificial active immunity, which we usually achieve through vaccination. You'll definitely want to know these categories. Live attenuated vaccines use genetically weakened microbes. Think of the smallpox vaccine. Killed vaccines, on the other hand, use heat or chemically dead pathogens. Then we've got mRNA vaccines, which actually trigger our own cells to make a specific protein to kickstart an immune response. And finally, toxoid vaccines. The trick here is that toxoid vaccines aren't made from the pathogen itself, but rather from the inactivated exotoxins that those pathogens secrete. Tetanus and diphtheria are your classic exam examples for that one.
And finally, Section 5, Clinical Case Patient Immunity. Let's see how well you've grasped this third line of defense, especially when things go wrong and it turns against the patient. Here's a high-stakes, life-threatening surgical complication to really test your clinical reasoning. Imagine you're in a procedure. Your patient receives mismatched donor blood, and they suddenly suffer a massive systemic reaction. What specific category of hypersensitivity is this? And what is the absolute first step the surgical team must take?
The answer is a hemolytic transfusion reaction. For your boards, you've got to know that this is a type 2 immediate hypersensitivity reaction. The patient's own IgG or IgM antibodies are actively, immediately attacking those mismatched donor blood cells. And the very first protocol-driven step the surgical team must take, you stop the transfusion immediately to save the patient's life. No hesitations.
Just to really cement this concept, let's look at the 2 absolute extremes of hypersensitivity reactions. Type I is your immediate, most dangerous reaction. We're talking anaphylactic shock here. It actively involves those IgE antibodies we discussed earlier, triggering a massive histamine release from mast cells. On the totally opposite side is type 4 hypersensitivity. This is a delayed reaction. It usually takes around 48 hours to even show up. The crucial exam point here. Type 4 does not involve antibodies at all. It relies purely on sensitized T cells secreting lymphokines, so organ rejection after an implant or a positive TB skin test, those are going to be your classic examples for type 4.
Okay, let's wrap this all together. We've mapped out the entire microscopic battlefield today, from the physical and chemical barriers of the first line to the rapid phagocytic and complement responders of the second line, all the way to the highly specific, memory-driven adaptive immunity of the third line. But here is why all this microscopic biology actually matters to your day-to-day responsibilities. Absolutely none of these natural defenses are 100% impenetrable. In the OR, your meticulous aseptic technique becomes the patient's ultimate safeguard.
So I want to leave you with this thought to mull over. When a patient's natural first line of defense is intentionally breached by the surgeon's scalpel, will your sterile technique be strong enough to stop those microscopic invaders from slipping in? Master these concepts, really trust your training, and stay vigilant out there. Good luck studying, and we'll catch you in the next explainer.